neurons. Okay, so if I'm talking about a multipolar resembles like that of any caudate nerve cells, beta caudate nerve cells, that is the nerve cells with a nucleus in the center, and it is going to have the dendrites on one side, a completely branched dendrites like that, and then it is going to have an axon, a long axon with the axonal ends. This goes and connects to the next dendrites of the other cell. So this is what you call them as the unipolar. This is a bipolar, and this is a multipolar kind of a stem cell. Multi because it is able to connect to many different neurons with the help of its dendrites. So that is called as multipolar neurons. Okay, so yeah, so that is what I was. Sorry, students, there was some problem over there. So, am I there?
Yes, ma'am.
Yeah, it's okay.
Yeah. Okay. For the next one, we are going to talk about the nerve cells. That is, this is a primitive nerve cell that we have talked about. Then you are having asexual reproduction. That is, there are two types of reproduction in the sponges.
Share screen, ma'am.
Yeah. It was still there on my screen, so I thought it is still there. Yeah. Is that fine?
Yes, ma'am. Thank you.
So you have two different types of reproduction, asexual and sexual reproduction. We talked about this type of reproduction even in the protozoans. Asexual reproduction, you have either by fragment. You either have a fragmentation, you have a budding type, and you have a formation of gemmules. We will talk about a little bit about this formation of gemmules in the next slides. And reduction bodies. So these are, fragmentation is when the body is broken into parts. That is called fragmentation type of reproduction. Budding. So this is the example of here. You have this budding over there, they bud off and then they are thrown out of the body, main body, and then this starts growing into a new one. So budding is one. We saw this type of a budding in protozoans also. And formation of gemmules. The formation of the gemmules occurs only when there is unfavorable condition. That is, if the conditions are not favorable for the poriferas to grow better, then a formation of gemmules. Gemmules is nothing but a small spore-like structures that occurs on the body, and these spores are broken out from the parent, and then they fall on the ground, and then they start growing up on their own. So that is formation of gemmules. And there are also type of sexual reproduction also occurring in the sponges. And this can be either, they also have a remarkable power of regeneration. This is such a beautiful topic, I can say. Power of regeneration is what the scientists are always very excited about, because can you understand what is regeneration? For example, you must have seen in the lizards, they chop off their tails, the tails are by autophagy, it is gone off, and then after some days you will find them regrowing, right? The same thing happens with the human liver. If the liver is broken or by accident, if you lose a part of the liver, we are able to grow them. But when it comes to different parts of the limbs in the humans, why are we not able to regrow that lost organ is a $1 million question. But here, when you're talking about this sponges over here, see, even if they are cut into so many pieces, each piece grows into a new sponge. Isn't it fascinating? When they can, why can't we? When we are chopped into two parts, why are we not able to grow the head part? Why is not able to grow the body, and the tail, the leg part? Why it's not able to grow uh, into a individual, complete individual? This type of example, if you remember, I'm going deviating a little bit from the topic. If you think talk about Ramayana, you can see the Ravana, whenever his head is chopped, he gets a new one, new head. Power of regeneration. When a Ravana can, why can't we? Though it's mythology, I'm not saying that it's something true. Even in that mythology, they were discussing about this points, is what I want to talk about. Okay. So power of regeneration. This porifera has this power of regeneration, and most sponges are hermaphrodite. I hope everybody knows what an hermaphrodite is. That is, if they are having both the male and the female reproductive organs in the same individual, then they are called as hermaphrodite. You can just make a note of it on the notes so that you can again go through it whenever you're having time. And they also exhibit protandry or protogyny.
Once again, explain the hermaphrodite.
Hermaphrodite means an individual, it can be either a male or a female, right? But here in sponges, both the reproductive organs are present in the same individual. Production of the sperms and also production of the egg is present in the same individual. So that is called as hermaphrodite, having both male and the female reproductive organs in a single individual is called as hermaphrodite. Okay. And they also exhibit protandry or protogyny. What is protandry over here? Protandry is the development of male reproductive organ first and then female reproductive organ is protandry. But if an animal or an individual has a protogyny, I'm talking about this only in a hermaphrodite. Okay, don't get confused with separate individuals. And when I'm talking about protogyny, gyne means female organs, gynecologists, that's how you call them. Talking about the female reproductive organs is called gynecologists. So protogyny, when the development of a female reproductive organ occurs first and then the male reproductive organs, then you call them as protogyny. So protandry, protogyny. So these are some scientific names you can just remember. And the spermatozoa arises primarily from the conocytes. Now you remember what a conocyte is. Where do you find the conocyte? On the conoderm, that is inside the cell walls of the poriferans. Egg arises from the archaeocytes or differential conocytes. Archaeocytes. Remember the archaeocytes? These are the totipotent cells, and these totipotent cells are the ones that give rise to new cells. Okay, so eggs arise from the archaeocytes, whereas the spermatozoa or the sperms arises from the conocytes. So this is the two different major places where the eggs and the sperms are formed. So here cross fertilization occurs over here. So fertilization over here, you can just have an idea about how they fertilize. So one, there is never self-fertilization. It is always a cross-fertilization. Even though they are hermaphrodite, it is always the cross-fertilization that is occurring. That is, a sperm is released from different sponges. That sperm swims outside. That is, they come out of the osculum, they swim outside, reaches into a new porifera, and they are being taken into the ostia. So this is, they are not going into the osculum. Remember, they are traveling only through the ostia or the pinacoderm. This osculum is only to throw out the things, never to accept. Okay, acceptance is only through the pores. That's why they're called poriferans. So the sperms are going inside the cell wall, and once they are inside the cell wall, the conocytes takes them and they pushes them inside. They take them over to an egg that is already present in the other sponge. and the fusion occurs over there. The larva develops. Once the larva is developed, there are 4 different types of larva that you find in the poriferans. We'll be talking about that also. Once the larva is developed, they are thrown out from the osculum, and this larva settles down on the floor and they develop into a new porifera. So that is the life cycle of the poriferans and cross fertilization. Coming to that is, these are some of the basic characters that we were talking about. So if they ask you to describe about the general characters of the poriferans, then you're going to talk about the pinacoderms, the conoderms, the pinocytes, the pinacocytes, the conocytes. What are the conocytes made up of? That whiz shape with a collar and a flagella. Then you're going to talk about the nutrition, the respiration, the excretion. Then you're going to talk about the spicules. Then you're going to talk about the canal, how the water enters and goes outside. Then you're going to talk about the fertilization, asexual or sexual type of fertilization. So if you're going to talk about this, then definitely you will get some marks for general characters of poriferans. Coming to classification, here we are going to talk the classification only up till class. So there are about 5,000 living species of sponges, including all those phylum Porifera, and all the species of this phylum are grouped into 3 classes. We are going to talk only about 3 classes, thankfully, not much are present. And this classes, 3 classes are divided into 11 or 12 orders, but we are going to talk only about classes. And they live up to a depth of 8,500 meters inside the sea. So that deep they are present. And they generally flourish in the warm waters. They don't like ice waters. And in the matter of size, they are highly variable. So each porifera, each class can vary in different sizes. So coming to the cleavage, cleavage is holoblastic cleavage, complete breakdown you find in this classification. And when I'm talking about classification over here, this pictures I just wanted to, when I was talking about this general characters, I wanted to have a talk about this. Here, when I was talking about the larval, different larval forms, these are the larval forms that I was talking about. When the cleavage is holoblastic, it includes 4 different types of larval formation. What are the 4 different types of larval formation? It can be coeloblastula larva, amphiblastula larva, parenchyma or parenchymula larva, or trichymella larva. So these are the 4 different larval forms that you find. If you can remember, it is well and good. If you don't remember, just talk about the cleavages, holoblastic type of a cleavage. So here, when I'm talking about a coeloblastic, coelo means coelom, some opening that is present is called as coelom. Holoblastula with flagellated cells. So if you can see this picture over here, this is a coeloblastula. There is a space in between, and all the whole surrounding is having a flagellated cells that are present over here. So whenever there is a larva of this type, then you're going to call them as coeloblastula larva. When talking about amphiblastula, amphi means both. It has both the characters. It's called amphiblastula, right? If you remember the amphibians, they can live both in land and water. That's why they're called as amphi, right? So when you're talking about amphiblastula, it is having 2 types of cell. So on the topmost, you have the flagellated cells. And when you're sorry, this is the picture, amphiblastula. So on the top, whenever you're going to talk about the uppermost part, it has the flagellated cell, and that is called the micromere cells, the minute cells. And at the bottom, it has large cells called the macromere cells, and a central blastocele present inside. So this type of a cell is called as amphiblastula larva. Amphi, both. It has both micro and macro cells. So that is called amphiblastula larva. Parenchyma larva, solid blastula with outer layers of flagellated cells. So this is a solid cell larva, which is having an outer layer of flagellated cells over there without any blastocele. That is what they're calling it as solid blastula. There is no blastocele as in the amphiblastula or a coeloblastula. Okay, so that is called parenchyma or parenchyma larva. Trichymella, you can find the flagella is just present on the equatorial region of the cell. Equatorial region is the central part of the cell, an imaginary central part over there. So if this is a cell with a solid blastula, that is even in this one, we don't have a blastocele, it's a solid blastula cells, but they have the flagellated cell only at the either sides. That is, you can also call them as north pole and south pole. But when you're going to invert the cell this way, then you're going to talk it about as equatorial cell. So it's better always to talk about equatorial cell. So these are the 4 different types of larval cells and porifera is going to have. And approximately 10,000 species known. So these are the different types of sponges that you come across in the seawater. So whatever the collections that you have, whenever you go to any seashore, you will find those vendors selling all different types of shells and all different types of sponges like this. So all these are the sponges that they go and capture them. Why they are solid? Because they are made up of calcareous spicules. Okay, so that is the reason why they are hard. When they die, they have become very hard. So the phyla is divided into 3 classes. We are talking about calcareae or calcispongiae, made up of calcium spicules, calcium carbonates. Hexactinellida or halospongia, they are made up of 6 rayed spicules. So they come under hexa. Hexa is 6. Tenellida is a race. So hexactinellida or halospongia and demospongia. This is another type of demospongia. So we are going to talk about 3 different classes: calcareae or calcispongia, hexactinellida or halospongia, and demospongia. And they are divided into 11 or 12 orders. So what are the different types? So here you can talk about phylum Porifera, divided into 3 classes: calcareae, demospongia, hexactinellida. And the examples that he has given over here, under calcareae, mostly you'll be talking about Leucosolenia or Scypha. Under demospongia, you'll be talking about Spongilla or Spongia. And under hexactinellida, we'll be talking about Euplectella or Halonema. In our museum, that is when you come for the practical classes to Ambedkar Open University, you will be seeing all these specimens. We have different types of specimens in our college, so you can just have a look at them. Did you start with your practicals?
No, ma'am.
No, ma'am.
No,
So-
madam.
Yeah, so when you are starting with your practicals, you will all will be coming to Ambedkar Open University at Banjara Hills, so
Yes.
Hyderabad. So there you can see different types of museum specimens. You can clearly see them and understand what are the different types. But till then, you can have a good look at the Google images. Fine. So yeah, so this is the picture that I was talking about over here: calcareae, hexactinella, and demospongia. And these are the orders that they are divided into. So phylum Porifera, class calcareae. So we'll talk a little bit about different classes. I'll just quickly go through those different classes so that we can talk about the canal system, because I'm very much worried about canal system. So class calcareae, exclusively marine, restricted to 100 meters depth, and requires a hard substratum for attachment. So they have to attach somewhere, right? Small size sponges, so you will find them at 10 centimeters in height. Cylindrical or vase-like structures. Osculum, very narrow. Remember osculum, that is the outer opening, and placed terminally at the end. Osculum provided with oscula fringes. So if there's osculum, something like this, an osculum over there, they have those fringes. So that is called the fringes, that is foldings over there, so that it can close and open whenever it wants them to open and close. Comparatively large collared cells. The collar cells are large. Skeleton represented by free calcareous spicules. And the spicules contain calcium carbonate 87% and 7% of magnesium carbonates. This can also be differentiated into megasclers and microsclers. Mega is a larger type of spicules, and micro is a smaller type of spicules. So, and then the canal system is, this is what I was talking about. Just make a note of it in your rough book. Canal system is Asconet type, Zygote type, and leuconoid type of canal system. So this is what I was talking about. Asconoid, Zygote, and leuconoid type of canal system. We will talk about each one of them separately. And what are the orders this class has? Homocoela and Heterocoela. So these are the two different orders, though we are not going to talk much about the orders over there. So these are the examples, and cycle. This is mostly what we'll be talking about in the canal system. Second one, hexactinellida. Hexa means 6. Actinellida, ray. 6 rays is called as hexactinellida, or it can also be called as glass sponge. That is, the sponges, they are very shiny, like they shine like a glass. So that is the reason why they are, the example is a glass sponge. They can also be called as glass sponge, halo spongia or glassy. Large size sponges on an average 10 to 30 centimeters in height. Wow. So almost a large scale that you can have, a big scale that you have. So a small scale to a big scale height is what this hexactinellida looks like. And they mainly survive in the deep waters, and between 200 meters to 1000 meters deep you find them. Usually cup or a vase or an urn shape like structure can be seen under this hexactinellida. So this is what the shapes I was talking about. The skeleton is made up of siliceous spicules or megasclers. So the big ones are the megasclers, and the small ones are the microsclers. And this is the shape I was talking about. Monaxon, single needle-like structure is the monaxon. Triaxon. So this is a triaxon. It can be of any shape like this. So this is a triaxon, and this is an hexaxon, though they are termed in the triaxon 3 plus 3. So hexactinellida also is placed at the triaxons. And tetraxon. Tetra means 4. 1, 2, 3, and 4. So 4 spicules you find. So they are termed as tetraxon. Polyaxon, if they are going to have many rays, then they are called as poly. Poly means many. So that is how all these are megasclers, whereas this tiny ones without any definite shape, all these are called as microsclers. Micro means small. Okay. Wall encloses a spongiocele, which opens by a wide osculum, is the same. And the canal system can be either Zygote or leuconoid type of a canal system. No dermal epithelium, and commonly called as glass sponge. Okay. So this also includes 2 different orders, Hexatrophora and Amphidiscophora. The examples over here, you have an Euplectella. This is a very beautiful. We have this in our college. You can have a look at this Euplectella. What do you call them? A specimen is present over there in a college. You also have a Halonema. Spironema we don't have. So these 2 we will have. So what exactly, why we are going to keep all these specimens in the colleges? Whichever the specimens that we are going to show in the college, you will have to read that. Only those will be kept for you all to display, and for your practical examination point of view. Whatever we have the specimen, you'll have to read it thoroughly so that you will have to be prepared for the exam. Okay, so the last one, Demospongia, people sponge. Why you call them as a people sponge? Demo means people. Spongia is sponge. People sponge. That is, in the earlier days, when we didn't have the synthetic sponge that we use in the kitchen, people used to use this sponge for working and to remove the water or something like that. Although we don't use it now, we have. We are all China mal made, right? Everything is use and throw mals we have. So synthetic ones are the ones that we are using now. Mostly marine, fewer fresh water or brackish water. I hope you know what a brackish water is, where the salt water and the river water mixes is called as a brackish water. this land is very fertile. Okay, so fertile land is where the brackish water is present. And 90% existing species fall under this class. So mostly whatever we are seeing right now in the corals or in the spongia, 90% comes under this class, Demospongia. Brilliant coloration. For example, the corals that you use, the pagadam antaru kada? Yeah, the pagadam comes from this sponges. So we use all those different color stones from these sponges. Okay, so skeleton either absent or siliceous. It can be siliceous, fibrous, and spongin fiber, or both can be present. Canal system. So they have a type of a canal system. What is the type of a canal system they have? They have the leuconite type of a canal system, and flagellated chambers, small or rounded. So this is the type of the larva that we're talking about. The larva, parenchyma larva, is what that is present in most of the demosponges. Freshwater species possesses contractile vacuole used for elimination of water from the cells. So these are the orders that belongs to Demospongia, and there are 7 orders that is present in the Demospongia, though we are not going to talk about them. Then this is the Demospongia examples over here. This is the Oscarilla, the red color, bright colored ones you can see over here, the Placina, the Spongilla. So that's why they we told that they resemble plant-like. Earlier, this sponges porifera were placed under plant kingdom, thinking that they are plant. But later on, when they came to know that they are not plants, so they were placed under a different phylum porifera. Cleona, the local ones, and the Microceona, another type of a sponge over here. So these are the example. Now coming to type study. So we completed general characters and classification. Now we'll be talking about the type study of Cycon. Phylum Porifera, class Calcareae, Heterocele, Cycetidae, family, and genus Cycon. Okay. So there's habit and habitat. Mostly you will find the cycons are being attached onto some hard substrate, or it can also be that is, they are being attached to some solid structures like a shell. So on a cell also you can find them attached, because they need some solid substratum to hold on to. So this is a shell that you can find, molluscan shells, and you can find the sponges attached on them. Okay, structure of the cycon, as I told, they can be anywhere between 5 to 10 centimeters long. So here they are branch cylinder structures up to 2.5 to 7.6 centimeters. And all the branches are connected at the base, and the base is always attached to some solid substratum. And they reveal, so these are that small dot structures. These are not some designs that they are given on the cycon, but they are the pores that you find on the cycon. So these pores are called as ostia, and the larger opening over here is called as osculum. So this is an osculum, the larger pore where the water enters. So this is a little bit of cut part over here that is given over there, how the ostia looks like. The water enters through this. This is the central cavity that we're talking about, or the spongocele. And from the spongocele, the water enters outside through osculum. So that opening is called as an osculum. The canal system, known as a cyconoid type of a canal system. And the canal system consists of paragastric cavity, or the gastral cavity, or the spongocele. So this is a spongocele I was talking about. And the wall of the cycon, they are always made up of the pinacoderms over there, or the pinacocytes. The osculum is surrounded by a large layer of cells called the myocytes, which are contractile in nature. Myocytes, when I'm talking about myocytes, I'm talking about muscle-like cells, which helps them in contraction and expansion. Whenever they want the water to be expelled out, they will open out, they expand, water is gone, and then they again close back, because no organism is allowed to enter inside the spongocele. It's only an efferent type. It has to throw it out, not getting it in. So myocytes are present over here near the osculum over there. So they act like a spinster, that is something like a valve which closes and opens, and they open through exterior, that is the osculum. So again, same thing. I don't have to talk much about it because I've already spoken about the structure in the general characters. I'll just go through a little bit fast over here. So this is the spicules that you find. Spicules is found in which layer? Mesohyl layer. So mesohylar layer is the one that is the blue color ones is the mesohylar layer. The conocytes is the conoderm, and the flattened cells all over the body is the pinacoderm. So these are the structure that you will have to remember, and you're going to draw this. If they're going to ask you to write type study of cycle, then you're going to draw the structure. You're going to give this, this opening is the ostium, that is the incurrent pore, which opens the water so that it can go inside. Then you have the spongiocele. So this is a spongiocele, and from here the water goes out through the osculum. That is an excurrent pore. Okay, coming to the radial canals. So, for example, if you check over here the cycle, here you have fold-like structures. Can you see these folds over here? So these are the fold-like structures that is present on the body of the cycle. Though the cycle over here seems to look like a solid cylinder, they have those large, the small pores all over the body, and these pores are thrown up like this fold-like structures. What are these fold-like structures? For example, I just made this paper cutting over here. Let me put it this way. So in this paper cutting, you can see the outgrowth that is found over here. So this outgrowths, this is called as a radial canal. This out one. So if you can see in the picture over there, so this is the picture that is a fold that you find. The same fold I've done over here. The outgrowth like this one is the radial canal, and the inside ones is the incurrent canal. You call this as incurrent canal, this as radial canal. And in this radial canal, you have small pores which leads the water inside, and from here it goes inside through the radial canal. The water comes from the incurrent chamber, goes through the pores inside, and then from here into the radial canal, and then they move into the spongiocele. And whatever the food that is being bought, they are taken up by this, and whatever the leftover excretion part is there, they are thrown out through the osculum. Understood this point now? Is this clear, this picture? So this is how the structure of a cycle looks like. So this is the folds that they have over there. And so if you can see in this picture over here, what I'm talking about, what is a radial canal? The outgrowth or the outpushings that you find over here, this outpushings, this one is the radial canal, and the gap in between the outpushings, this one is the incurrent canal. And what do you find in a radial canal? You have this prosopiles are present in the incurrent canal. What is a prosopile? So you can see over here, this is an incurrent canal, and this opening is called as a prosopile. Make a note of these points in your rough book. First thing, talk about radial canal. What is a radial canal? The outpushing is a radial canal. The inside ones, the tubular-like structure is the incurrent canal. The openings of the incurrent canal is called as the prosopiles. This opens inside the radial canal, right? And the radial canal has this conocytes. What's the help of a conocyte? It helps in pushing the water current. They pull the water current inside, and they push whatever that is present. The microorganisms is also brought about inside this water current. And from here, they go into a narrow opening called apopyle, A-P-O-P-Y-L-E, apopyle. So a narrow opening inside the radial canal is called an apopyle. So water, understood this? Radial canal, incurrent canal, prosopiles, apopiles, and excurrent canal. So you have to understand all this canal system. Fine. Let me tell you once again. The outgrowth is the radial canal. So this is a radial canal. And after the radial canal, the inside like this in pushing, which is towards the outside, outer environment. So this is the in-current canal. The in-current canal is having an ostia-like openings, and this opening is called as a prosopile. This prosopile opens inside through a narrow opening towards the gastric cavity or the internal cavity through a pore called apopyle. From the apopyle, the water goes out. Comes in. You can't say it goes out. It is coming in. So water comes inside the spongocele over here, and from the spongocele, the water is pushed out through the osculum. That is the outgoing water. So this is what you will have to understand about this different canals over here. Any doubt over here in the canals? Radial canal in current canals. Any doubt? If you don't have doubt, I'm just going to go to the next.
Ma'am, prosopile.
Prosopile, yeah. If you can see in this picture, prosopile. See this picture over here, that black and white picture over there. Can you see that?
Yes.
This is the mirror image of this picture over here. So this here, it is the inside one over here, the radial canal. So let me tell you over here. What is this one? This is the radial canal, right?
Yes.
This opening is the in-current
In-current.
canal. Very good. So this is an in-current, this is a radial canal.
Yes.
Where do you find the prosopile? Prosopile is the opening from the in-current canal inside the body cavity of the cycle. Something like a gate, the main gate which opens. The water has to come, goes into the prosopile. From the prosopile, it goes into the radial canal. From the prosopile, they go into the radial canal. From the radial canal, there is a small, minute opening over here called the apopiles. Prosopiles, apopiles. Fine?
Yes. Yes, ma'am.
So remember, as something like prosopiles are the main gate, right? And apopiles are your door, house door. They have to open them from the gate. They have to come inside, and then when you open the door of your house, only then they can come inside. So remember it like that. Prosopiles are the outside gate, and apopiles are your house
Inside.
house door. Fine. This is a gate, and this is a door. Remember that.
Okay.
Fine. Understood now?
Yes, ma'am.
Yeah. So these are the prosopiles, apopiles, and the radial canal and the in-current canal. So that is what I was going to talk about. So here, when I'm going to talk about the X-current canal, when I'm in-current canal, X-current canal, don't get confused. What is an in-current and an X-current canal? In-current is water when it is coming from outside, it is called an in-current canal. X-current, X means it has to go outside. So X-current is when the current is being produced inside the walls of the spongiocele, the water is moving away. So when they are moving away, then you call them as X-current canal. Got the point? In current is you're asking the water to come inside. X-current is when you're asking the water to go out. Is X-current canal.
Exit.
Fine. Exit. Exactly. Yeah. Good.
Yeah.
So that is about X-current canal and the in-current canal. And now you know what an apopyle is. This picture will tell you all what are the different types of cells that are present in the cycle. The same thing that I spoke about. So what is this layer called? Anyone? There are, you can see the conocytes over here. So when the conocytes are present, what is the dermal layer of the conocyte called as?
Conodome.
Very good, conoderm. And who's that? Saida. And then you have a flat cell. What is this cell called? Saida, can you please mute your system? Fine, thank you. Yeah, so this is flat cells. What is this layer called as? Pinaco. conoderm. So this layer is a pinacoderm. This layer is called as conoderm. And what is the central layer over here? Mesohellar cells. Fine. So physiology of the water current. So what is exactly happening over here? From the in-current canal, the water enters the in-current canal. From the in-current canal, it goes into the prosopiles. From the prosopile, they enter the radial canal. From the radial canal, they enter the apopyle. From the apopyle, they go into the excurrent canal. From the excurrent canal, they are going into the spongiocele. From the spongiocele, they are going outside through the osculum. So ostia or osculum over here. So if you can see in this picture over here, this is an in-current canal. Water is entering an in-current canal through a prosopile. Into the prosopile, they go into the radial canal. From the radial canal, they go into the apopyle. So you have an apopyle over here. From the apopyle, they are pushed out as excurrent canal. From the spongiocele, they go out through the osculum. So this is the water current system or water canal of the poriferans. The cycons, skeleton of the cycon, I've already talked about the cycon, so you can just go through this pictures of here, different types and shapes of here, the monaxons, the tetraxons, the triaxons, and the polyaxons over there. Physiology of the water current, I've already spoken about the physiology. I'm not going to go into this. Go through this, and if you have any doubt, you can just ask me, because everything is the same. I was talking about this particular topic I've already talked about. And nutrition, respiration. What is a nutrition? Whenever the water is coming inside, the water brings inside microorganisms. Whenever there's a microorganism coming, the conocytes engulf them and pass them to the amoeboid cells situated below the conocytes. As you can see in this picture over here, whenever the water is coming inside through the pores, the conocytes, so these are the conocytes, they engulf the food particles and they push them below. So can you see this amoeboid cells? They are pushing or passing the food to the amoeboid cells that is present in the mesohellar region of its body surface. And there the digestion occurs. The amoeboid cells is going to take the food, assimilate them, holozoic type of nutrition, assimilate them, and it is conveyed to various parts of the parts of the porifera or the cycle. Nutrition, holozoic. I have talked about this holozoic type of nutrition and saprozoic type of nutrition. Holo means complete devouring of the food is holozoic. Saprozoic is if they are going to taking in the liquid part of the food is called a saprozoic type of nutrition. But here the cycon has an holozoic type of nutrition. Digestion is intracellular, which is compared to that of a protozoan. That's what I was talking about. And respiration and excretion takes through body diffusion. Again, diffusion on the surface, whatever taking in of oxygen, and excretion is through vacuoles. Excretion is done in that way, and respiration is by diffusion. And reproduction. So we'll talk about a little bit of the reproduction. Asexual type of reproduction. They can be either budding, fragmenting, or gemule formation. So when there is any outgrowth like this in a body organism, they grow a little bit and then they are chopped off. So there's cut off from here, and this goes and lands on a solid substratum and they start growing. So that is the reproduction of asexual reproduction. What's happening in a sexual reproduction? We also talked about this. The sperm from a different cycon comes. It is thrown out from the osculum. They enter into another cycon through an ostia over here. So there are the pores. These are carried, the conocytes carry the sperms over there into the mesohellar region. And once into the mesohellar region over here, they are carried to an egg. The fertilization occurs over there in the mesohellar region. Dividing of the cell occurs over here until the formation of the larva. Once the larva is formed, what are the different types of larva that we talked about? Any one person, if you have written it in your rough book, can you tell me all the 4 different types of larva that we studied?
Coeloblastula, amphiblastula,
larva,
trichemella,
larva, parenchyma larva, trichemella larva.
Very good, very good. So see, that is the importance of making a note of it in a rough book so that you'll be able to tell it very fast. Otherwise, what happens? We will have to recollect what all we studied. So once the larva is formed, they are put into the spongiocele, that is the gastric cavity over here. From the spongiocele, they are thrown out by the osculum. And once outside, the free swimming larva goes and attaches itself onto a substratum, and they start growing into a new sponge. That is sexual reproduction. So this is what the picture is. When we are talking about asexual reproduction, it can be budding, it can be fragmentation, and also I said it can be by gemule formation. So here I'm going to talk about a little bit about this gemule formation, or the gemmules that is present. So these gemmules are internal budding. Can you see those yellow spots on this cycle? So this yellow spot, that is whenever there is any unfavorable condition. So when I'm talking about any unfavorable condition, it can be because of high temperature in the water, or because of low temperature in the water, or if there is any toxic substance that have been mixed in the water, so that also can be unfavorable condition. Or if the cycon is not able to survive anymore further, so what they do is they try to produce the small gemule-like structures, these yellow spots on the body. How do they look like at a closer look? They look like a round structure. That's why it's gems. You know, the gems that we eat, everybody loves them, right? The gems, it's in the shape of that round shape, and the round shape is covered or made up of the spicules of here. The spicules can be of different types. It can be a monaxon, tetraxon, or different types. It has a micropyle opening, and inside is the archaeocytes or archaeocytes. What is the function of an archaeocyte? One word. One word. What is an archaeocyte? What is a character of an archaeocyte?
It's a totipotent cell.
Very good. Very good. Totipotent. That is, they can give rise to new cells. So this is the cell. Once it detaches from the body of the mother cell, that is the cycon, adult cycon, they fall into the ground, and this archaeocytes is going to grow into a new cycon. So that is how the gemule formation is all about. So if they ask you to write in a short answer, describe the gemule type of reproduction, then you're going to talk about this type of reproduction. And when does this reproduction occurs? Only during unfavorable condition. Okay. And the sexual reproduction I've already told. So the sperm cell does not enter the ovum directly. The union is assisted by the conocytes, and the conocytes help them to bring them to the egg cells, and the fusion occurs. That is sexual reproduction. Okay, development of cycon. So what is happening over here? Fertilization of the egg into a zygote. The zygote divides repeatedly to form a round mass, the blastoderm mass, mostly covered with homogeneous cells. Homo means same type of cell, homogeneous. Hetero means different types. Here it is homogeneous type of cells. But at one end, a few thick granulated cells appear, and homogeneous cells grow into flagella. This is the larval form that we talked about. One side it is the flagella type, and on the other side it's a solid blasto type of cell, blasto cells that are found over there. So what is happening over here? So when a zygote is formed over here, just concentrate on the picture over here. When a zygote is formed, they undergo cell divisions, and once the cell division is undergoing, they have some flagellated cells on one side, and they also have solid cells, the megasclers or megameres, and this is a micromere cells. All the micromere cells have flagella, and the megamere cells have large cell-like structures. And once the cell division is occurred and invagination occurs over here, what is happening over here? It has amphiblastula larva is formed over here. Amphiblastula means double. That is, it is having two types of cells over here. One is the microcytes and one is the megacytes over here. And once this microcytes are being formed with the flagella, what happens is this is released from the osculum of the mother, and they are released into the water. Once they are released into the water, what is happening over here? They go and attach to a substratum onto the flagellated side. Okay, so they are going to go upside down. So you can see the arrow over here. They have put it as an upside down. The flagellate is going to get attached to the substratum. Once it is going to attach to the substratum, invagination occurs. So they are going to invaginate something like, if I have to talk about the invagination, if this is a stick-like structure, they are going to invaginate and they're going to become like this. So they're going to become like that. Okay. So invagination is what is occurring over here. So they are going to bend like that. So once the invagination occurs, I can't put it in. These are some things that has to be done on the blackboard, but never mind. See, they are going to bend like this, invaginate like that. Got this point now? Invagination, right? So they're going to bend like that. And once they are bending like that, what is happening is all the flagellated cells are going to go inside, and all the mega cells are going to be on the dermal layer. The outer dermal layer is formed by this mega cells, and the micro cells are the ones that is going to go inside the spongoceles with the conocytes. And this conocytes are the one that is going to help in water current to go inside and throw them outside. So this is the development, further development of larva into an adult form. Canal system. So that was about the type study of cycle. Hope you have understood that. Now we are going to come to another important. Do we have time? Can we take another 10 minutes time? If you all are okay. Do you all have another class?
No, ma'am, you can take it.
Can I? Okay. This is a very important topic, as I was telling you all. Just pay attention and give me some more time so that I can complete this canal system over here. Note all the important points in the rough book, please. And if at all you all are having any doubts, get back to me. Okay, so just ask me to explain once again so that I'll be able to explain to you all. This question you will be asked for major question, so please pay attention for this type of canal system. So canal system, I told we're going to talk about 3 types of canal system. And in this 3 types, the leuconoid is again divided into sub systems. What are they? We will talk about that. So one is the Asconet type. Asconet type is a simple cell division. So simple cells division is an Asconet type of a canal system. Then you have a Zygote type of a canal system. Zygote type, we've already talked about the radial canal, something like this. So this is the Zygote type of a canal system. And understand the blue color ones are the pinacocytes. There's a pinacoderm layer. The red ones are the conocytes. And then you have a gray color that is a mesohyl. The gray color ones is the mesohyl layer. So these are the 3 layers that is common in all the type of the cycles: Asconet type, Zygote type, and leuconoid type of canal system. So what is that? We will talk one by one. First, we'll talk about Asconet type of canal system. Asconet type of canal system is the simplest canal system. So what is happening over here? First, understand this layers over here. The outer layer. What is not coming, ma?
Now it's okay, ma'am.
Fine. Okay. So this is the example of leucocyllina. So example of a leucocyllina, which is an asconoid type of a canal system. The outer layer is a pinacoderm. The pinacoderm is innervated by the incurrent canals over here, or the radial canals, or it is also called as ostia, the dermal ostia. Ostia is the pore. So a simple ostia is present over there. This opens into the conocytes. This is the conocyte cells, conodome or the conocytes, and you have this conocyte cells, which is a collar cells, you call them. They are all having those flagellated cells over here. With the help of this conocytes, the water current is being, because of the movement of the flagella, what is happening is the water from outside is pushed inside, and from here it goes into the spongocele over here. From the spongocele, whatever the microorganism is being brought about from the water current, they are captured by the conocytes and they are thrown inside the mesohyla cells for further digestion. and also the oxygen is diffused into the dermal layer of this cell, and the excretion is done from the spongiocele or the gastric cavity. From here, whatever the waste is present is being thrown out through the osculum. So whenever the water has to be thrown out, only then this osculum opens, otherwise they are closed. Because what I told, they have a type of a cell. What is the type of the cell that they have in this osculum? The type of the cell is called as myocytes. Myocytes, nothing but something like it resembles like that of our human muscles. Okay, so because of that muscles present, myocytes present, they are always closed. Only when they have to throw the water, they will open up, and the whole water goes outside through osculum. So this is an Asconet type. What are they? They just have a simple dermal ostia. The water comes inside through the dermal ostia, and the conocytes are present in the conoderm layer. The conocytes capture the food, and also the diffusion of oxygen occurs over there, and whatever waste matter is there is excreted with the help of the movement of the flagella that is done by this conocytes. So that is a simple asconoid type of canal system. Okay, so here I've given you all the points. You can just go through the points to understand. This is nothing but whatever I told you all. Now coming to this is also another type of example of here, how they look like. So this is a picture I told you all. Now this is what the specimen looks like. The example of the specimen over here is Olynthus over here, a vase, the flower vase shape Olynthus. You can see all these dark dots. So these are the ostia, dermal ostia, and you can see the triaxon spicules, the spicules that are present, and they have cut open to show how the spongiocele looks like, and this is the osculum from where the water goes out. So this is how the picture. For every water current, if you remember this picture, this arrow shapes ones, you'll be able to write this answer very well. Water from the exterior goes into the incurrent pores. From the incurrent pores, they go into spongiocele. From the spongiocele to the osculum. From the osculum, they are out. That is the Asconet type of canal system. Example, Olynthus and Reproduction. Now coming to the next one, Zygote type of canal system. Zygote is again easy for you because we have already talked about it. So these are, once again, last and final I'm telling us, this is the radial canal, and this is the incurrent canal. And from the incurrent canal, there is an opening. What is this opening called? This opening is called the prosopiles. Water from the incurrent canal goes into the prosopiles, that is the dermal ostia. From the dermal ostia into the prosopiles. From the prosopiles, they go inside the radial canal. From the radial canal, with the help of the conocytes, when the flagella is moving, the water is being pushed into the apopiles over here. So this layer is an apopile over here. From the apopile, they go into the external canal. From the external canal, they are in the spongiocele. From the spongiocele, the water goes out.
Why is it not clear, ma'am?
Okay, my mouthpiece is gone. Yeah, sorry. Is it clear now?
Yes, ma'am.
Right. Did you miss anything over here, or it's fine? Did you understand?
Fine, ma'am.
Fine. Okay. So this is a Zygote type of canal system. So Zygote type, what all you have to remember? You have to remember that there is an incurrent canal. So this is an incurrent canal. This is a radial canal. Then this is the prosopiles, the apopiles, the spongiocele, and the osculum. So remember this, you will be able to write this answer very well. So this is the points that you will have to remember, and you can write, use this for your answer. Read it and you can write them. So in the same way, if you remember this gist, you will be able to understand, that is, from the dermal pores, the water enters the incurrent canal. From the
previous page open.
This one?
Yes,
Or
ma'am.
this one. I'll be sending this to you all in a PDF format, so don't worry about this. You will only have to
Which group, ma'am?
didn't you all get the protozoan PDF?
No, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am, we got.
You all got it. I don't know why you didn't get. Who is that who didn't get it? Please ask your friends to resend to you so that you can just go through that. All this will be given to you all, so don't worry about writing the points. I want you all to only write important points. Okay, so this answers you can just go through when I send this picture.
Please send group open university group. PDF.
No, I am not into that.
No, ma'am. I am asking with my
Your
classmates.
friends. Okay, you just talk about that after the class.
Okay.
You can just think on that points. Okay, fine. So coming to the picture of your the top points, dermal pores. From the dermal pores goes into the incurrent canals, from the incurrent to prosopiles, from the prosopiles to radial, radial to internal, that is apopyle. From the apopyle, it goes to the spongiocele, osculum, and outside. So that is the, what type of canal system is this?
Psychonite.
Very good. Psychonite type of canal system. Saida. Is that Saida who spoke with me?
Yes, ma'am.
You are very active in my class. Thank you, Ma Saida. Okay.
Thank you, ma'am.
So coming to the last type of a canal system, so this is the leuconide type of a canal system. Leuconide type of a canal system is little bit complicated, but definitely not very hard, because they are after all very small organisms. Doesn't have to take much time. Okay, coming to this leuconide type of a canal system. See now, here the chambers are made up of globular or round structures over here, unlike the one that we found over here. So this is some waves like structure in the psychonite type of a canal system, whereas in leuconite type of a canal system, you have some chambers. That is, all those have been separated as chambers over here, and they are round chambers that are present over here. But the cells are all going to be the same. So you have this pinacoderm or the pinacocytes that are present outside. They open into a dermal ostia. From the dermal ostia, the water is going inside. And then inside they have this conocyte chambers. Then they open into a prosopiles. From the prosopiles, they are opening into the spongiocele through an apopyle. So all these canals that are opening into the spongiocele, all these points over here. So all these are apopiles over here. The rest of the ones over here, all those you find over here, that is interconnected with different chambers. All these are the prosopiles that we'll be talking about. And from the prosopile, they go inside this chamber. From the chamber, they go and open into the apopiles. From the apopile, they go into the spongiocele. From the spongiocele, the water is thrown out through an osculum. So simple. What you have to do is, only in a leuconite type of a canal system, you'll have to make some round chamber like structures. And then you can say that this is a leuconite type. All these chambers should be interconnected on all the sides, on all the four sides. They have to be interconnected, and then it will be easy for you all to draw this diagram. So simple leuconite type of a canal system. Examples of this type is Spongilla or Spongilla, Spongio. This is a diagrammatic format, and this is what exactly it looks in a live spongia, the dermal pores. And these chambers, the round chambers that you found over here, this type of a chamber over here resembles something like this over there. So this is the picture of two different types, that is Spongilla and a Euspongia. Any doubt in this? From the ostia, the dermal ostia, the water is entering into the incurrent canal. From the incurrent canal, it goes into the prosopiles. From the prosopile, they open into another chamber. From there, the conocytes help the water to be thrown out into the apopiles. From the apopiles, they are going into the spongiocele. From the spongiocele, after the food has been captured, the whole thing is thrown outside through osculum. So that is the leuconoid type of canal system. So the water course of here, dermal ostia, incurrent, prosodus, if present. The prosodus and apodus are again different types of names given to different sponges. So you don't have to get tensed up. It's only a type of gateway, that before prosopiles you have a prosodus, and before apopiles you have an apodus type of currents over there. So from there, the water goes into a large channel, that is a spongiocele, from there to oscula, and then outside. So this is the different types of canal system, that is the Asconet type, Syconoid type, and Leuconoid type of canal system. So here I was talking about the Leuconoid type of canal system exhibits 3 different stages of evolution. So before they became a proper leuconoid, they showed some different stages of evolution. Under this evolution, you'll be talking about Eurypylus, aphodal, and diplodal type of canal systems over here. What are there? I'll just go through quickly. What are they? So here, if you can see, Eurypylus pilus type of a canal system, I'm going to talk very shortly. So here, the chambers, because we have already talked that these chambers are round in shape. So this is what the round shapes you find in this type of Eurypylus type of canal system also. So this is a timble shape, that club shape. Timble is, if you remember, when you're stitching something with your needle, you cover your finger with some round cap-like structure. Does anyone do stitching at home? Or if you have seen a tailor?
Yeah, ma'am. Yeah, ma'am.
Yeah, they have that cap-like structure so that the needle doesn't poke their fingers. So they just have a cover cap-like structure. So that is called a timble. That is a shape over here. So this is timble shape here. Notice this diagram over here. This timble is not closed like in leuconoid over here. All these chambers are closed, single round structures over here. But when you are going to talk about is Eurypylus, they are all open. All these round chambers are
Small openings like
open.
ostia.
Yeah, they are not separate. They are open. So that this
Yeah.
shape resembles like that of a timble. So that is why it's called as a timble shaped openings. So what is happening over here? The same way, the water from the dermal pores or the ostia enters into the subdermal space, a small dermal space over here, subdermal space. From there, they go into the incurrent canal. So they pass into the incurrent canal like that. And then from there, they go into the prosopiles. So the opening over here, the small opening is a prosopiles over there. From the prosopiles, the flagellated chambers push the water into the apopiles. And from the apopiles, they go into the excurrent canal. And from the excurrent canal, they go into the spongiocele. And from the spongiocele, they are out of the cycle through the osculum. So this is the Eurypylus type. So when you're talking about the Eurypylus type, don't forget to mention the timble shaped chambers that are present in this type of the cycle. When you're coming to aphodal type of canal system over here, so what is happening over here? A simple one. The chambers are connected with an incurrent, a large incurrent over here. From there, the incurrent is opening into small chambers. From the small chambers, they open into the apopiles, and from there, they are opening into the excurrent canal, and from the excurrent canal, they open into the spongiocele, and then the water goes out. So dermal ostia, subdermal spaces. So you have some subdermal spaces over here. Then into the prosopiles, from the prosopile into the chambers to aphodas or apopiles if present, and then from the apopiles they go into the spongiocele out to the oscula. So that is aphodal type of a canal system. And a diploidal type of a diplodal type of a canal system, again almost same like that of an aphodal type of a canal system over here. Only difference is they are being this incurrent canals are a bit larger in this type of diploidal type. In some cases, there are also narrow current tube, very narrow current tubes are also present. The prosodus is between the incurrent and the flagellated chambers. In current canal and the flagellated chambers, you have a prosodus. That is what we talked about in the leuconoid type of canal system. Just before the prosopile, you have a prosodus, and then just after the apopiles, you have an aphodus type of a canal system present over here. The same thing, you're going to talk about this. And this type of a canal system you find in Spongilla and Oscarella. Mostly, they will ask you to talk about the 3 important canal system, which are there. That is the Sycon type, the Asconet type, and the Leuconoid type of a canal system. And when you're talking about the leuconoid type of canal system, just mention that they are divided again into 3 different types. That is this diploidal, then you have the aphodal and the uripilus type of a canal system. Don't go much into the detail, but we're going to talk only about the important points. Then you have one more Ragan type. Ragan type, when you're doing this practicals, you will be coming across this type of a canal system, Ragan type. This is also again in the cycle only. What is the specialty over here is they resemble a pyramid shape or a triangle shape of the structure over here. Very simple structure. They have a base hypophyre, which is not having any ostia. The ostia is only present on the body surface like this, and the water enters the ostia through the flagellated chambers, and from the flagellated chamber into the apopiles, and into the apopiles to spongiocele and the osculum. These chambers are what is enlarged when we do a cross section of this particular spongiocele or the sponge. You have this particular shape over here, something like lungs, right? They resemble something like a lung. So this is a dermal pores. From the dermal pores, the water enters through this incurrent canals. Then from the chambers, the flagellated chambers, the water is going inside the prosodus. From the prosodus into this chambers, small flagellated chambers. From there into the apopiles or the aphodus. From the aphodus into the apopiles, and then they go into the spongiocele. So this is a Ragan type of a canal system. You might be asked this in maybe some MCQ questions. What is a Ragan type of, do sponges have a Ragan type of a canal system? So you should be able to remember that there is something called Ragan type of a canal system. So this is the points over there that you'll have to remember. Functions. What are, so when we are talking about this canal system, what are the functions? Why should something like this be present in this type of sponges? So these are the functions. What are they? They provide nutrition, they provide respiration, excretion, and reproduction is done in the canal system. And they also bring in food. That is, nutrition means they're going to bring in food. Respiration means they're going to get in oxygen. And excretion means they're going to take away the carbon dioxide and nitrogenous waste from the body. And for reproduction, they're going to carry the sperms, and then they're going to fertilize the ova and throw the larva out through the spongiocele so that they form into a new one. So this is all about the phylum Porifera, and I just given you all this picture so that it'll be easy for you all to understand. So this is all about the phylum Porifera. Hope you people have at least understood 50% of what we spoke over here. Yeah, was that good enough? Did you all understand, ma?
Yes, ma'am. It's good. Very interesting.
Zoology itself is so interesting, you know.
Yeah.
You can just keep talking more and more on that. Anyways, because there's some
Yes.
time constraint.
Ma'am,
Any
can you
doubts?
send the PDF, ma'am? Which
Yeah,
group
tell me.
will be send? You will be send.
I think there is 2 groups over there, BSc, BZC something, and
BSc Open University.
BSc.
I am there, ma'am, but I can't receive.
Okay, so you're not an admin over there. All those who haven't got it, you please take my phone number so that you can add my number into your group, so that whatever I'm sending, I'm going to send it to you all also. Or at least one person be common in your group. Why is that you people have 3-4 groups when you all are of the same batch?
Ma'am, we are a same batch, but we are not in same center. So we don't know.
Okay.
Don't
So that
know
is
about each other.
okay. Fine. Then in that case, just take down my number so that you WhatsApp, just say a hi, hello. And
One
then
group is in chat box. Please click the link and join the group, ma'am.
I'm very bad at joining the group. I don't know how to. How do I?
Ma'am,
I'm not a big fan of
link is there, ma'am.
Yes, ma'am.
Link is there in a chat box.
Where is that link now? No.
Link is there, ma'am, in a chat box.
No, I don't find any links over here.
In chat box, ma'am.
Yeah, I don't find any link. So yeah, I didn't find any.
More option, ma'am. There you can
Ma'am,
find.
please click more. Ma'am, click more.
Okay,
Yeah.
more. Yeah,
Yeah.
more. And then post.
No, in chat box it is not there.
That's what.
Chat
I don't have
is
any
in
link.
chat
So
First you send the link, then ma'am will
I
click
know,
to
that's
the
what
link.
I'm
No?
Prayas Thakkar name. Send the link to.
okay.
Give your
Forget
contact
about
number, ma'am. They will add you.
No,
Give your
I'll-
contact number. They will
that's what.
add you.
That's what. Please take down my contact number. Whoever has not got this PDF.
Yes, ma'am.
9490
9490
93
93
74
74
22.
22.
Yeah, I'm Dr. Padmini over here. So you can
Yes,
just
ma'am.
say hi, and then I can send the PDF to you. You can send it to your group. Any one person.
Please do forward in the group also,
Thank you,
girls.
ma'am.
Definitely.
ma'am.
I'll forward. Okay, ma. See you all tomorrow. Thank you.
Thank you, ma'am.
Ma'am, what
Same
about
time?
records, ma'am?
What about?
Records?
Did you all start with your practicals?
No.
No, ma'am.
When you're starting your practicals, then you worry. No, now you
Ma'am,
start.
records will be there.
Yeah, definitely. You will have.
It will be provided when?
Provided like what? The record books or what?
Record books and
Uh,
nothing is told about
material.
records and practicals, no information at all.
Manual book and record book, ma'am.
they will give you all the information. Record books you will have to buy. If at all anybody is in contact with the seniors, you can
Madam,
just yes, ma.
With the material, they sent some laboratory manual and record books, madam.
Record books like your
Like
physics and
textbooks,
chemistry.
they are like textbooks only.
Only textbook. When you start coming to the college, you will have to buy a practical record book, the notes book. In that, you will start drawing or writing something.
Management will say when we will come in practicals, they will provide us material, practical material
Okay. Then you can
at
do
that time only.
Yeah. So what you can do is, all this will be the same. Whatever you're doing in the theory, you will have a little bit in detail in the practicals. Whatever the general characters we are talking over here, remember that your life will be easy in the practicals. That's what we can say, right?
Okay.
Ma'am, how many units we are going to have for first semester?
How many units? Whatever the syllabus that they have given to you all those you will be having up till
Yes, 12 units.
echinoderms.
12 units, ma'am.
All the units, yes, up till echinoderms.
Till
Huh.
hemichordate.
Hemichordates, yes. Hemichordates is a very small topic. Echinoderm, up till echinoderm, it's a big topic. Hemichordate is just one page answer. You can just go through that. Nothing much about that. Yeah.
So all the 12 units we are going to have for first semester, right, ma'am?
Obviously, everything is important. All that we will be touching a little bit, not the whole portion. See, if you're going to talk about the whole thing that is present in this universe, this 6 months is not enough for us. You need a lifetime to understand all those. So everything, one example we'll be touching everything, because as a zoologist, we should know how from a single cell we have become this multicellular and the specialized organ, because the humans are the if not very powerful, but at least they are very intelligent. So intelligent is what always wins more than power, right? So how we have evolved this much, how our brain grew, we have to understand. For that, we'll have to understand unicellular structures. Only then we will talk about the multicellular, and under multicellular also properly developed chordates. So how all this occurred, we'll have to understand. Only then, if you learn, only then we will know what zoology is all about. Yeah. So don't take your degree as a big burden. Read it in a way that you are learning something new, you're assimilating some new things that you come across in this universe.
Yeah, it is very interesting to learn visits.
Yeah. So if you think it in that way, then definitely zoology is going to be very fascinating and interesting.
Yes,
Yep.
it is.
Yeah.
Yeah.
Okay,
Your
so
explanation way is very nice, ma'am.
thank you.
Thank you.
Any doubts? You can just keep asking.
No,
I
ma'am.
will try to explain it. to you.
Only please send
We
me
will
PDF.
get informed, na, ma'am. We will get informed, na.
About
Records and the
yeah.
practicals.
Everything will be told. Everything will be told in the.
Yeah.
When, ma'am?
No, this much time got.
All those points, I think Mr. Kishore will be able to answer you all because he is the counselor over there. Hope you know who's Mr. Kishore over there.
No, ma'am.
No, no, ma'am. We don't know.
Zoology department. Zoology department. What you can do is one day you can just go to the department, go and talk to them. You have the counselors in Department of Zoology. All your questions will be answered by them. You have Dr. Purna Srinivas sir, and you have also Dr. Kishore sir. You can go there, talk to them, and they will let you know every details. Okay.
Okay, ma'am.
Okay.
Thank you, ma'am.
Okay, ma. See you tomorrow then. Okay. Bye.
Thank you,
Okay.
ma'am. Bye.
Thank you, ma'am.
Thank you, madam.